Dr. Anna Lee, who comes to us from the University of Minnesota, and she's going to speak to us about untangling the molecular mechanisms of nicotine and alcohol co-addiction.
All right. Thank you very much for inviting me to participate in this conference. It's been a wonderful morning so far. Just put this here. So I'm going to switch gears a little bit and present some data that I've been working on looking at alcohol and nicotine co-addiction and take a slightly more molecular tack on, as a pharmacologist, always trying to identify drug targets that we could use to treat co-addiction. So alcohol and nicotine, again, are the two of the most commonly abused substances. Co-use of these two drugs is also very common, and so I just want to bring people up to date on some of the current statistics for alcohol and cigarette smoking. For this crowd and for all of the clinicians out there, I probably don't need to go into this quite very much, but the national cigarette smoking rate is about 16%, and this has actually dropped quite a bit over the last 10, 15 years. But there are still some differences in the cigarette smoking rate amongst different demographics. So those that have no high school education, below the poverty level, self-report psychological distress, have a higher cigarette smoking rate than the national average. And cigarette smoking is still a very costly disease in terms of tobacco-related diseases. It costs $125 billion US per year for these healthcare costs. For alcohol, alcohol has actually increased over the last 30 years. So the national average alcohol use disorder prevalence has risen to 13%. And also I wanted to highlight this additional factor, high-risk drinking. Now, the people who exhibit alcohol use disorders don't necessarily have high-risk drinking, vice versa, but high-risk drinking has gotten a little bit of press over the last year because of its increase over the last decade. So high-risk drinking has increased 30% over the last 10 years to 13% nationally. The largest increases were seen in specific demographics. So there was a 60% increase in high-risk drinking amongst women. So amongst elderly, those 65 years and older, high-risk drinking increased 65%. Minorities and low socioeconomic status also showed increases in high-risk drinking. And high-risk drinking was defined as drinking, exceeding the daily drinking limits at least weekly during the prior 12 months. And that's 5 drinks a day for men, 4 drinks a day for women. So exceeding that every week for the last year, essentially. The co-occurrence of alcohol and nicotine has been tracked over the years, and a lot of the data that looked at this was collected in the early 1990s, and this data was mostly collected in men, but it does indicate that there is a high co-use of these two drugs. So almost 90% of heavy drinkers were smokers, and so we're really interested in looking at co-addiction, co-use of these substances, alcohol and nicotine together, which is probably the norm rather than the exception. So in my lab, I really liked to take a look at this in sort of a shared and separate mechanisms manner. So if you take a look at this circle diagram here, we're really interested in seeing, okay, what are the molecular mechanisms in this blue circle that influence alcohol use? What are the ones that influence nicotine use? And what are the things in the middle here? And in my lab, we're really interested in taking a look at genes and molecules that are important for it, taking that out to the behavior, and then back again. And so I just wanted to mention that in the last couple of years, we've tried to investigate this, in a more behavioral level. How do we get voluntary co-consumption of alcohol and nicotine in mice? Mice is what I work with. And so I've published a couple of papers there. I don't have time to talk about that work for this presentation, but I'd be happy to talk about it afterwards. And then for today, I'm going to present a story that's been going on for a while in my lab, looking at a particular molecule, PKCE epsilon, and how it is important in alcohol and nicotine consumption and reward, and its role in regulating nicotinic receptors. So for this crowd, I really don't need to go into a lot of background on these nicotinic receptors. I just wanted to highlight that the complexity in these receptor systems really come from its subunit combinations. And so it was mentioned briefly before, but there are a lot of different subtypes that have been discovered. New ones are being discovered every day. And the importance of stoichiometry is also apparent, because for each subtype and probably each stoichiometry of each subtype, the receptors have different activation profiles, different ligand binding affinities, different expression pattern differences, different rates of desensitization. And so I just wanted to put up this picture that illustrates some of the expression profile differences that we see for the different nicotinic receptor subunits. And so this is an in situ hybridization figure from one of Sharon Grady's papers. And you can see the diversity in expression pattern. This is a coronal section in mouse brain. So for today, I want to talk about two of these subunits, alpha 4 and alpha 6. These, and I just wanted to show here the expression pattern differences of alpha 4, its very common subunit partner, beta 2, and then alpha 6 and beta 3. So alpha 4 and alpha 6 have been associated with alcohol and nicotine addiction in human genetic studies, as well as in mouse studies where the genes themselves have been deleted. And so you have these alpha 4 and alpha 6 gene knockout animals that show altered alcohol and nicotine addiction related behaviors, such as consumption and reward. So keeping that in mind, I'm going to tell you a story about a molecular conversion point of alcohol and nicotine that touches on these two subunits. So PKCE epsilon is a kinase that I started working on in my postdoc, and you know, most people here probably don't know about it. It's a part of the serine threonine kinase family, this 9-isozyme family. The vast majority of substrates for PKCE epsilon are unknown. So we do know that it phosphorylates the TRPV1 channels, it phosphorylates NaV1.8, and I'll show you a little bit of data that it phosphorylates the nicotinic acetylcholine receptors. PKCE is very widely expressed throughout the brain and the periphery. It's been shown to be involved in alcohol addiction, in neuropathic pain, in stroke, in cancer. And in this diagram, there's PKCE epsilon. It does not express a calcium binding domain, so it's not activated by calcium, but it is activated by other lipid second messengers. And so in my postdoc with Dr. Bob Messing, they were really interested in looking at the role of PKCE epsilon in alcohol addiction-related behaviors. And so they had these PKCE epsilon knockout mice, and they had quite a bit of work demonstrating how PKCE epsilon was important for alcohol consumption. And so this is a typical type of study. This is an alcohol 2-bottle choice where the mice are individually housed. They receive 1 bottle of alcohol, 1 bottle of water in their cages. And you can see here that the knockout mice drink less alcohol than the PKCE epsilon wild-type animals. And there was some data in the literature that suggested PKCE epsilon was also important for nicotine. And so I ran sort of similar studies using a nicotine 2-bottle choice with PKCE epsilon knockout mice, and showed that these animals drink less nicotine compared to the wild-type animals in blue. Now, these changes in nicotine consumption, nicotine and alcohol consumption, are associated with differences and reductions in alcohol and nicotine reward as measured by conditioned place preference. So here you can see with the wild-type animals, they develop preference for a chamber where they have been repeatedly conditioned with 1 gram per kilogram of alcohol. Knockout mice don't show any such preference to that chamber. Similarly, PKCE epsilon wild-type animals develop preference for 0.05 mg per kg nicotine, whereas the knockout mice, again, don't show any such preference. So this is an interesting finding because this is a molecule that, or a kinase that is seemingly important in both alcohol and nicotine addiction behaviors. And I just wanted to highlight that this is, the PKCE epsilon knockout animals don't show like a pan decrease in motivation or reward. So other drugs of abuse like morphine, there's no decrease in morphine self-administration in these animals, no differences in cocaine-induced dopamine release in the striatum, and no differences in sweet consumption. And so this suggested that PKCE epsilon may be a point of molecular conversion in alcohol and nicotine addiction mechanisms. So what are these mechanisms? How do we get at this? This is an overview slide of the data that we have found so far. So just to summarize, quickly right now, we found that PKC epsilon regulates alpha-4 and alpha-6. And again, these are the two subunits that are important for alcohol and nicotine addiction. We found that PKC regulated alpha-4 and alpha-6 through two different molecular mechanisms. And I'm going to go through a short vignette of the data for each mechanism before moving on to some unpublished work. But we found that PKC epsilon phosphorylates the alpha-4 subunit, affecting alpha-4 beta-2 nicotinic receptor function. And we also found that it reduces the expression of alpha-6 and beta-3 mRNA levels, again affecting receptor function. And combined, we think that the PKC epsilon knockout animals are showing this deficit in alcohol and nicotine consumption reward because of these mechanisms. So starting off with the first side of the story, one of the interesting things about nicotinic receptors is their ability to be desensitized for prolonged periods of time and then to recover from the desensitization. And so this is just a very, much abbreviated cartoon of the different states that the receptor can be in. And using whole cell electrophysiology in hex cells that express the human alpha-4 beta-2 nicotinic receptor, we can see this desensitization and recovery. So here, acetylcholine, puffs of acetylcholine will cause activation of the receptor. And then if you bath apply acetylcholine onto the cells for 60 seconds, this will desensitize the receptors. After washout of this acetylcholine, if you get, again, test for receptor activation, you can see the receptors slowly recover back to baseline. And this recovery curve is affected by PKC epsilon activity. So if we plot this recovery curve here, so after desensitization, you wash off of the acetylcholine, and then you just assess for recovery of receptor activity under normal conditions in black. When we inhibit PKC epsilon activity in these hex cells, as shown in the blue dotted line, we see a very different recovery curve. So somehow these receptors don't recover back to 100%. And so the curious thing about this was that previous data in the literature had suggested that this was mediated by PKCs, but they didn't identify which isozyme of PKC was important. And so what we thought, well PKC epsilon's, it phosphorylates things. Let's see if it phosphorylates the alpha-4 subunit and mediates this effect. So we took a look at the intracellular loop of the alpha-4 subunit. So the loop between transmembrane domains 3 and 4 is a large loop that contains a lot of regulatory elements. We subcloned this loop and then incubated it with and without PKC epsilon, and then ran mass spectrometry to identify phosphorylation sites in vitro. And we found 5 PKC epsilon phosphorylation sites in this loop. And 4 out of the 5 sites are conserved in humans, mice, and rats, suggesting that these sites may be quite important in receptor function. So to identify how important this was, we mutated each one of these sites from a serine or threonine to an alanine, and then expressed that mutant alpha-4 subunit with a wild-type beta-2 subunit in hex cells and ran the same desensitization experiment. And 4 out of the 5 sites showed a decrease or this deficit in resensitization recovery. I'm only showing 3 on screen right now. But you can see that some of the mutations, so S465 to alanine, S467 to alanine, almost completely recapitulated what we saw in the cells when we inhibited PKC epsilon activity. And so all of this data was collected in cells. And one of the things we wanted to do then was to see, is this site or are these sites phosphorylated in vivo? And we moved on to generate a phosphoantibody against S467. And so here, this is just, again, testing in cells, the detection of the wild-type alpha-4 subunit, the mutant alpha-4 subunit with the S467 to alanine mutation here, and there's really no detection. And we assessed whole brain synaptosomes in PKC epsilon wild-type and knockout animals, and there was a small but significant decrease in phosphorylation of S467 in PKC epsilon knockout mice. So this suggests that, yes, this is phosphorylated in vivo. The second thing is that this is likely not the only kinase to phosphorylate this site. This site is also a suspected PKA phosphorylation site. And third, we believe that the accumulation of these phosphorylation deficits over the 5 sites in the alpha-4 subunit may be causing quite a effect on the receptor itself, recapitulating what we saw with PKC epsilon inhibition. So now how does this relate to the reduction in alcohol and nicotine consumption and reward? So the idea is if alpha-4 and alpha-6, or alpha-4 is required or is important for alcohol and nicotine consumption and reward, and in PKC epsilon knockout mice, these mice have deficits in receptor recovery, there may be less overall receptors to be activated or to be fully activated in these animals, leading to reduction in the effect of alcohol and nicotine. So that's the first sort of tidbit, story. The next story, we started off in vivo instead of in vitro, and so we looked at the rationale for starting this way, looking at mRNA levels, was that we didn't really have very good nicotinic receptor antibodies. That's just not available. And so we wanted to make sure that there were no changes in mRNA levels by doing an mRNA screen between PKC epsilon wild type and knockout mice to ensure there were no differences in mRNA levels. And lo and behold, we actually found something. So the PKC epsilon knockout mice had a very interesting decrease in alpha-6 and beta-3 mRNA levels in the striatum and the ventral midbrain. And there were no elevations or any changes in any of the other nicotinic receptor subunits that we looked at, including alpha-4 and beta-2. So I wanted to just take a moment and talk about alpha-6 and beta-3. You'll notice that the decrease in mRNA was almost exactly the same between the 2 subunits and exactly the same between the 2 brain regions, the striatum and the ventral midbrain. And you can see here, this is alpha-6 and beta-3, very heavily expressed in the VTA and the substantia nigra, as well as the terminals of the dopamine neurons that project out to the striatum. Now, the genes that encode alpha-6 and beta-3 are found back to back, and this suggests that these 2 genes are being co-regulated. And so, because we had seen a decrease in mRNA levels, we wanted to see if this translated into any changes in receptor function. And so one of the well-known and documented functions of the alpha-6 beta-3 nicotinic receptor is its role in regulating dopamine release out in the striatum. And this phenomenon has been well described by the Craig lab. So I just want to go over this a little bit. And so this is your striatum. The dopamine neurons project from the VTA into this region. And also there are cholinergic interneurons that are tonically active in this region, and they're releasing acetylcholine onto these alpha-6 beta-3 beta-2 containing nicotinic receptors. When this happens, there is a frequency-dependent depression of dopamine release, so that depending on the, tonic versus phasic firing of the dopamine neuron, you're not actually seeing that much of a difference in the dopamine that's released from the terminals, depending on whether it's single pulse or burst firing, as it's described in this diagram, or you can think of it as tonic versus phasic firing. Now, under conditions of reward, when reward happens, these cholinergic interneurons turn off, and you can artificially mimic this by antagonizing these alpha-6 beta-2 beta-3 containing nicotinic receptors. What happens in this situation is that this relieves the depression, so that now tonic firing gives you a certain amount of dopamine release, and phasic firing of these dopamine neurons gives you much more dopamine release. And this is thought of as a salient signal for the animal to pay attention to its environment because something important is happening. And this is dependent on alpha-6 containing nicotinic receptors. And so we took a look at this phenomenon using voltammetry. And this is in wild-type animals, so under normal conditions, you don't really see that much of a difference, about a 50% difference in the dopamine release between single pulse versus burst pulse, or tonic versus phasic stimulation of the dopamine terminals. When you inhibit the alpha-6 containing nicotinic receptors with alpha-conotoxin M2, this now expands to about 225%, so this is your salient signal. This signal is impaired in the PKC epsilon knockout animals. And so we think that this impairment of salience is, again, another reason why these animals show decreased alcohol and nicotine consumption and reward. So for a while, the story ended right there, but somebody had mentioned sex differences, and all of the data that I've shown you so far and all of the data that has been collected with PKCE epsilon and alcohol have all been in male animals. And so when I got the animals imported into the lab, of course we started breeding them, of course we generate females, so we tested the female animals and we have come up with a very different story. And so most of this work was done by my graduate student, Jana Moen. And we wanted to first see, what does the alcohol and nicotine consumption phenotypes look like in PKCE epsilon wild type and knockout females. So for females, we actually don't see a reduction in alcohol consumption until the concentration goes out to 20%. Whereas in the male animals, you can see here it starts to separate at 4%. So you can say, well, they still have the same type of phenotype, it's just not as strong. For the nicotine consumption data, we really didn't see what we saw with the males. And so with the male animals here, you can see this, remind you that this is the decrease that we see in consumption. This is what we see with the animals here. And so we really didn't see any difference in nicotine consumption weeks 2, 3, and 4. If anything, we saw a slight increase in nicotine consumption during week 1 in the PKCE epsilon female knockout animals. And then again, if I can remind you, this is what we see for alpha-6 and beta-3 mRNA levels in the males. This is what we see in the females. So nothing out in the striatum, no changes in mRNA levels, and an increase in the transcript for alpha-6 and beta-3 in the ventral midbrain. And this increase, again, was almost exactly the same between the two genes, suggesting they are co-regulated, but they're co-regulated differently in females than they are in males. And so we wanted to see if we can confirm this increase in alpha-6 activity using a different behavior. So we turned towards the alpha-6 hypersensitive line of mice. These are transgenic mice that have a point mutation in the alpha-6 subunit that renders them hypersensitive to agonist. So they will be activated at levels of agonist that don't activate the wild-type alpha-6 receptor. And in these animals, if you give them a low dose of nicotine, they will show this nicotine-induced locomotor activity that you don't see in wild-type animals. We see the same type of thing in the PKCE epsilon female knockout animals, this increase in locomotor activity at a low dose in the female knockouts that you don't see in the PKCE epsilon wild-type females. Again, suggesting that these mice may actually have increased alpha-6 nicotinic receptor activity. And we've been sort of racking up interesting sex by genotype differences in behavior that we're just trying to wrap our brains around. And so I'm showing this slide because I'm hoping that somebody can help me understand it. So nicotinic receptors are important for depression, and they've been implicated in depression. Nicotinic receptor antagonists like mecamylamine and partial agonists like cytosine have antidepressant-like effects in mice. And so this is a tail suspension test that we ran between PKCE epsilon wild type and knockout males and females. And the greater the immobility time, the more depressed the interpretation is that the animals have a more depressive phenotype. So there's no differences in untreated male or female PKCE epsilon wild types or knockouts in their baseline levels of activity in this test. Desipramine, which is our positive control, it's an norepinephrine reuptake inhibitor, works just as well in male and female wild types and knockouts. But then varenicline, which is an alpha-6 and alpha-4 partial agonist, appears to work only in the females, knockouts. And so we're not quite sure how to interpret this data. Perhaps varenicline only works because you need a higher level of alpha-6 for the antidepressive effect to show up. I'm not quite sure about that. So just to summarize, we see a really interesting sex difference in PKCE epsilon regulation of alpha-6 and beta-3 mRNA levels and activity. Ultimately, we think that PKCE epsilon is a really interesting point of molecular conversion for alcohol and nicotine consumption and reward. So future directions, we're really trying to now figure out what this sex difference is. So we know that one of the upstream activators of PKCE epsilon is mGluR5. And we know that there are extranuclear estrogen receptors that also work with mGluR5 mGluR5 to activate specific signaling mechanisms in female animals. And so we're going to try to take a look at that and see if that's different between these animals and whether or not that's one of the underlying causes of the difference in alcohol and nicotine consumption and reward. So with that, I'd like to thank the people in the lab. Everybody who is in italics is an undergraduate in my lab. They have been absolutely phenomenal. And I want to thank my current funding from nIChRs and NIDA, as well as my past support. And I'd be happy to take any questions.
Thank you for a wonderful talk, bringing together two big drugs of addiction, tobacco and alcohol. And Jed.
Thanks very much, Anna. That's really interesting. And I guess one question, though, that jumped to my brain was the alpha-6 antagonist, alpha-6 containing, subtype containing receptor antagonists, they increased dopamine release in the striatum, if I understood correctly. And I'm just trying to understand behaviorally what that would mean, because one would think that for nicotine to be rewarding, does that mean that nicotine is desensitizing alpha-6 containing receptors and that will provoke dopamine release?
Yeah, so that's a really, what I can say is that the role for the alpha-6 receptors out in those terminals is like a unique function. So I sort of think of it as alpha-6 role in the soma of these neurons versus the terminals of these neurons. So alpha-6 activation in the soma of these neurons may increase nicotine or increase neural activity and increase the propensity for these dopamine neurons to fire. What we're looking at the striatum is just the role of these receptors in gating this frequency-dependent dopamine release. That is a function of the cholinergic interneurons in this region. So yes, if you turn off the cholinergic interneurons, and these neurons are inhibited by GABA projections during conditions of reward, that has the effect of enhancing this frequency-dependent dopamine release. You can just artificially reproduce that with an alpha-6 antagonist. But what an alpha-6 antagonist does at the striatum may not necessarily translate to what it does, on the soma of the neuron, and then even, in general, on other, like the total activity of it. That was just one of our methods of really isolating whether or not there was any differences in function in these genotypes.
And I'm not surprised Al has a question about alcohol.
Dr.
and
Lee,
nicotine.
you really don't know me unless...
I remember you, Al,
okay.
from SR&T.
We did about 30 years of work in the area that you're involved in. And my first response is, wow, I'm really impressed. Good job.
Thank you.
I have some things that you need to think about. One of them was going to make you go back to your days with Rachel. In 1976, oh boy, is that a long time ago, Pat Hatchell and I published our first paper on nicotine, and it was nicotine metabolism in 3 mouse strains. And the girls metabolized the nicotine faster than the boys. And I use those terms because they're relatively young mice, 50 to 60 days of age, and we call those adult. But if we did the same thing in humans, we'd be talking about boys and girls. But nonetheless, consistently over 30, 35 years of studies in multiple mouse strains, the females metabolize nicotine faster than males. So in your species comparisons, I think you've got to put that in your noodle and sort that out a little bit. With respect to the alpha-6 and beta-3 mRNA levels, I squirmed, and I'm squirming again because of a number of reasons. First off, we showed in multiple studies with upregulation of receptors that you don't change messenger RNA. But more relevant to your studies, probably if you dig around in the Mike Marks or Sharon Grady's papers, you'll see there were studies that were done with the heterozygous mutants that have half the binding, but the mRNA didn't go in the same direction. So equating messenger RNA with protein expression really makes me nervous.
Yeah.
I would strongly recommend that you talk to Mike McIntosh or Paul Whiteacre. Either or both. Probably Paul can actually do some conotoxin binding for you and sort out whether you actually have changes in receptor expression.
Okay.
But great work.
Thank you. So I'd like to address both of those points. We did take a look at nicotine clearance in the male PKCE wild types versus knockouts, straight up injection of nicotine and then sacrificing the animals at different time points and running HPLC. There's no difference in nicotine clearance in the males. In the females, we didn't have access to HPLC, but we did take a look at CYP2A5, so going back to the Rachel days. There's no differences in CYP2A5 expression in female wild type versus knockout PKCE mice. We haven't directly compared male and female, so that's, yeah. And then for the conotoxin binding, yes, mRNA does not necessarily equate to protein. And in fact, we were very cautious because in these animals, the mRNA levels were in naive, so they weren't nicotine treated. This is just what the animals have. And so we then, tried to really look at function. We have partnered with Jerry Stitzel to take a look at some conotoxin binding between male and female wild types and knockouts, and the results are confusing. So it looks like we see no changes in the ventral striatum for conotoxin binding in either genotype or either sex, but we see a small difference, a small drop in conotoxin binding out in the striatum, in the nucleus accumbens specifically, in both male and female knockouts, not wild types. So how does that, how do you jive those two pieces of data? What we're thinking of is that perhaps the conotoxin is reading out binding of fully formed receptors that are at, on the surface of the cell or functional. And maybe what we're reading for the mRNA levels is also supported by some of the functional studies that we do, maybe looking at different time points in the maturity of these receptors. So maybe we're looking at mRNA levels, the amount of immature and amount of receptor subunits that are being shuttled up to the surface. So yeah, those two pieces of data are disparate, and we're trying to wrap our brains around it.
You do have to remember that the messenger RNA that we measure with in situ hybridization studies, so the figure that you put on the screen there, that came from my lab probably 20 years ago now.
Mm-hmm.
We're measuring principally mRNA in the cell body. And the vast majority, at least as best as we can tell, of the alpha-6 containing receptors are expressed in the nerve terminals and the modulating, as you showed, the amount of release that's generated by an action potential. By the way, before I forget, the vast majority of receptors that we've measured in mouse that are alpha-6 containing are alpha-4, alpha-6, beta-2, beta-3. They comprise 60 to 70% of the alpha-6 containing receptors. So the model that you use, alpha-6, beta-2, beta-3, they're kind of low man on the totem pole, so I worry about that.
Thank
Okay,
you.
I think we need to move on. Wonderful talk, wonderful discussion. We will have more time for discussion at the end of the morning session and again in the afternoon. Thank you.